And we're going to start. We wanted to interview Miss Joanna Gibson, who lots of you know. She's a superstar in our watershed. Joanna. I will tell you, so her former boss happens to be in the audience, so I won't tell you how long I worked on getting her to come over to the water agency, but it was a significant amount of time to get Joanna to come and work for us. And because I knew about her talent and who she is as a person, her character, her values, and her just really deep understanding of our watershed. So, Joanna, how'd you get your start? What's your origin story?
So my mother used to kick us out of the house in the morning, and we weren't allowed to come back until it got dark. So I spent a lot of time outside. I did my undergraduate degree in Australia. I worked in the lab during university counting macrobenthic invertebrates. And after that, I got a job with the university for a year or two. Then I moved to New South Wales and worked for the New South Wales National Parks and Wildlife Service. And looking back now, I'm actually really surprised I got that position. I used to go into that office every week and say, do you have any work? I was unemployed at the time. And then somebody on the field crew actually dropped out. They weren't available to go, and they remembered the annoying person who kept coming into the office every week. So that, it was a contract position for several years, but we were working on a comprehensive regional assessment in northeastern New South Wales, where we were looking at, were the national parks large enough for the wildlife species that were on the threatened and endangered species list? Do we need more national parks in different areas? So I think that's where I got my flavor for conservation work initially. Then skipping forward multiple numbers of years, I did my master's degree at Purdue University in northern Indiana. And then after that, I've bounced around with the US Forest Service, Audubon, CDFW for 10 years, thank you, Jeff Brandt, and Karen Cleary-Rose and Nick Peterson, if he's in the audience as well, were on my interview panel I worked with the Western Riverside County MS-HCP on the biological monitoring program. And then Heather pulled me over into the water district for the HCP.
When she says pulled over, what she means is I didn't stop calling her. So Joanna, how long have you been working in the Santa Ana River watershed?
What year is it? Probably nearly 20 years now, I think. It's close to it, about 16 years.
I think I've been in it for 16 years as well. I remember the first time I met Joanna was out in the field after there was a problem with the permit compliance for the East Branch Extension pipeline across the Santa Ana River. I was with U.S. Fish and Wildlife Service at the time. She was with CDFW. And I remember, I was thinking, who is that tiny Australian lady? She's so cool. And here we are all these years later, probably like 15 or 14 years later, sitting here in front of everybody today. Okay, so I want to ask you all the things that everyone wants to know. What part of Australia are you from?
I'm from central Queensland, a really little tiny town that nobody's heard of called Yeppoon, Yeppoon. Okay. There was no fast food restaurants, no traffic lights. There might be a traffic light there now. Lots of roundabouts.
How did you get to the United States?
I actually, after I've been working for New South Wales National Parks and Wildlife Service, a lot of Australians take about a year off and go travelling around the world. So I did that, and I thought it would be a great idea to do some work in Oregon. So there's an organization called the Student Conservation Association that's based over here in the States, and they take people from around the world and from in the US and place you with different organizations. So I was placed with the Forest Service in Oregon on the Umpqua National Forest. And then I got a full scholarship to do my master's degree at Purdue, and I've been here ever since.
And what did you study in your master's program?
I looked at, this is one of my subjects on the screen right now. I looked at the effects of prescribed fire on eastern box turtles. I actually worked on a military base, Army military base, to do my field work. So I have lots of funny stories there. I'd have to check in at the gunnery range every morning, and the worst day was getting a call asking, "Ma'am, where are you today? We're firing the tanks. We've got the tanks out on the range, and we forgot you were out there." Another one, I used to regularly to run into the guys that were training out there. This was all map and compass back then, and they weren't sure where they were, so I'd help them out with their maps. And then another group that asked me what I was doing, and then he said, "You hear that, men? If you see a turtle with an IED on its back, don't fire at it."
So how would you compare, well, let me ask it a different way. Did your master's work adequately prepare you for the Santa Ana River?
I think it partially did. So it was conservation bent, and it was also involving land management or land management recommendations. Definitely a lot of research, data analysis, all of that goes into hand in hand with furthering your career in biology anyway.
So we have quite a few students in the room. I was at this other conference this morning, and there was a slide that said, "Do you really need to write a master's thesis?" What's your answer, Joanna? Do you think everyone should write a master's thesis?
Yes, it's painful. It can take a long time. Mine was very large. But there's AI now.
Yes, very true. So for our students out there, our fellows that I all see sitting right there, when you get into graduate school and they ask you, do you want to be in a program that writes a master's thesis, you should say yes, because you will get extraordinary knowledge and understanding, not just for your subject matter that you're studying, but also how to write in a way that you can convey your thoughts, which you may need to do someday. I can't tell you how many staff memos we write to our board, and every time I do, I go back to that master's thesis work, and I think, oh, thank goodness I did that. So don't skip that fun step of graduate school. So what was your graduate degree in, specifically?
Ecology.
Ecology, okay. And so then how did you decide what career path you were going to go on? Because that's kind of a broad degree.
Well, first of all, it was getting an income. That was important. So after that, I did do field work. Different people that I'd met over the years, I'd reached out to them and asked them, did they have any positions on their field crews? So I was in Arizona for a little while doing field work there. And then a position came up actually in Orange County at Star Ranch Sanctuary, which is an Audubon Society property, 4,000 acres, and I worked there for a couple of years. And I was actually still finishing up my master's thesis then, like finishing up writing it. At that point in time, the economy was really bad, but I did actually, having 12 years of experience and a master's degree, started with CDFW as a scientific aide, because that was all that was available at the time, and I was willing to do anything. So that's when I got picked up with a biological monitoring program, and I was with them for just a year. And then I think I had a phone conversation with Jeff Brandt and shifted over to the Ontario office in Habcon and spent the next 10 years working on a lot of different projects in the watershed. A lot of CEQA, streambed agreements, and then the last 2 years we're working on the Western Riverside and the Coachella Valley MSHCPs.
So tell us a little bit about the work that you're doing now in the Upper Santa Ana River Habitat Conservation Plan that, yes, is still out there. We're still working diligently towards our permit. Joanna has us this close. Brian Croft is in the audience. I see you back there, the chief at Fish and Wildlife Service. So tell us a little bit about that. That was why I needed you to come over. When I got this job, one of the board's questions was, how are you going to replace yourself leading the effort on the Habitat Conservation Plan, which, in case you're not familiar, is a gigantic regional plan that permits over 100 different water projects for the next 50 years. We've been working on it since 2013-14. I remember when we, since he's in the audience, Jeff's getting a lot of love today. I remember when we said, you know what, we're going to expand it so we can cover all of our water's impacts and also, figure out all of our mitigation for these projects for all the permits that we need. And Jeff Brandt said, that's going to take you an extra 10 years and $10 million. And it turns out, Jeff, you were right. So well done, Jeff, for calling that. But Joanna's still working on it, and it actually is an amazing example of proactive conservation permitting and planning in a way that you can protect the health and vitality of a watershed while also producing the different infrastructure products that we need to have our functioning, society and communities here. And since I got this job 6 years ago, Joanna has been leading that charge. So tell us about how you feel on a daily basis, where we are, and kind of the value of this effort that you've been putting forth for 6 years now on behalf of many, many agencies in our watershed.
So I have a really large brown paper bag that I breathe into regularly. This is a really big effort, and it does take a lot of time, and it's hard for people to understand that. We are working with different agencies who are understaffed and underfunded. So trying to move things forward can feel like it's very slow. But this is a comprehensive strategy where we are putting forward mitigation up front, getting it on the ground so that we don't have what's called temporal loss. And honestly, without it, I don't think things, especially for Santa Ana Sucker, would be changing or getting any better. So it's kind of counterintuitive. You need the projects coming forward that are providing the funding that are going to help implement the mitigation strategies, at least for that species, that are needed. So the plan area is 863,000 acres. We have 22 species that are covered under it, 21 of which will be impacted. A lot of it is based on habitat that's been modeled. Obviously, you can't go out and do on-the-ground surveys over 863,000 acres for 22 different species, especially when you don't know exactly what the project footprint's going to be yet. But this is a comprehensive strategy, a lot of mitigation being put forward up front, and it will provide the vehicle or the strategy for projects to come online. And it's not, once we get the incidental take permit and the BO from the US Fish and Wildlife Service, that's not carte blanche, we're off to the races and we can go out and do all the projects that are in there. It means that we have a streamlined process in place. So each project still needs to come in and demonstrate that they are consistent with the impacts that we identified and that we have the mitigation in place ahead of time for that project for it to go forward.
Is that one of the turtles with the bomb on its back?
Oh, yes. So on the right-hand side, eastern box turtle, so I had to put transmitters on them, and working by yourself with trying to hold on to something, mixing epoxy, and getting that in place and not getting it all over yourself or the turtle running off, I came up with this turtle stick. No turtles were harmed. It was a 2x2, and I would duct tape them on the top of it and hold it between my knees, and then I was able to mix the epoxy and get the transmitter put on the back. And then the photo on the left, these guys are one of my favorites. This is a stink pot turtle, or a musk turtle. They look like a taco, I think. And true story, they can be on branches or on trees several feet off the ground. So they often fall into canoes. If you're going underneath, they'll just randomly drop into the boat.
That's really good experience for you to have now on the Santa Ana River.
Exactly.
That is awesome. So what do you think the value is, going back to the Habitat Conservation Plan? And I remember when I first got to U.S. Fish and Wildlife Service here in 2010, I worked on the recovery plan for Santa Ana Sucker, and I remember reading it and being like, this is all great, except for we're never going to have the money to actually do most of these things. And now, 16 years later, we're actually implementing the majority of the conservation measures and activities that are in the recovery plan for the Santa Ana Sucker, which is extraordinary. And the cool thing is that it's done by and in partnership with the water agencies from all over this region, which I don't think is what Fish and Wildlife Service had envisioned when they were writing that recovery plan. But Joanna is so amazing. They're actively working on construction of restoration of Anza Creek and Hidden Valley Creek in the Riverside areas, which are 2 recovery actions listed in that recovery plan. Joanna was able to get out, I think, like $9 million in grant money. We have the contractors out there right now. We're working with Dustin and Riverside County Rangers on helping us with all the vegetation management. So the cool thing about this is that it brings everybody together to work on one plan of action in a way that no one agency could have done it themselves. Certainly not U.S. Fish and Wildlife Service, certainly not CDFW. So as a conservation biologist, which I think is what you are at heart, what do you think the impact of that is, and like the impact of you personally in this watershed to be able to move those things forward?
Well, I'm one part of a large team. It's a we effort, and it couldn't happen without you and our board. But we are definitely moving things forward, I think, in a positive direction. Everybody brings something to the table, so the 11 water agencies that are part of the HCP effort, some have money, some have water, some have land. So by bringing it all together, we're getting a more holistic landscape scale approach, which is what you want. I'd say one thing that I found to be very frustrating at CDFW is that if you're in the permitting arm of things, by the time the project hits your desk, no changes can be made, essentially. They're 100% at, 100% design. You're not going to, the applicant and the engineer is not going to be entertaining you suggesting, can we make some changes here? And then the mitigation strategy is piecemeal. It's applying a Band-Aid after the fact. So in the HCP, like for Hidden Valley Creek and Anza Creek, so Anza has some hydrology right now. It's fed by 2 storm drain outlets, so it is independent on what I lovingly call urban slobber. And Hidden Valley Creek actually has no hydrology at all. So those projects, it's larger than just the construction of them. So we will be, when we get our permits, redistributing water that is currently 100% discharged from an outfall at the Riverside treatment plant and shifting it to 5 different tributaries. So we'll be able to provide year-round hydrology, which will provide additional habitat for Santa Ana sucker to get up and out of the main stem river into the tributaries. And then, along with that is the host of other species that are found in those areas. So hopefully western pond turtles and all of our riparian riparian-dependent birds. So that's a larger comprehensive effort than that, one agency wouldn't have been able to pull off by themselves, for sure.
Definitely. And I think like one of the impacts that I'm really proud of is when we first started in, I think it was about 2010, 2009, as we started working towards trying to take on this habitat conservation plan, there was very little science that was done on the populations of the Santa Ana River, specifically the native fishes. We had the RCRCD, Kerwin and Brett were out there, taking samples here and there, but it wasn't like a concerted effort. And so that's when in 2015, when we really started to take on the habitat conservation plan, that fortunately my board was very supportive when I said we really need the USGS to design and implement a comprehensive native fishes survey. So we started on that in 2015. And I think, sitting in on a few of the talks today, and Brock from USGS is here, and he publishes all the time. The body of work on the Santa Ana River is extraordinary now compared to, in the early 2000s. And to see the knowledge and understanding that has grown over the years is just really fantastic. And I'm super proud of that part of it, that we were able to invest. And fortunately, thanks to the support of our board, we were able to invest in high quality, defensible science that furthers understanding, so that then when we make restoration decisions or management decisions, we have something that we can lean on that really, really helps. I think my mic is having a problem. So Joanna, tell us what your favorite species are while they get my mic fixed.
Horn lizard for sure. I felt bad, but I did make one squirt blood out of its eye because I was harassing it until
Okay.
I caught it.
For the non-scientists in the room, I think you should probably explain that.
So they have a defensive mechanism to try and get predators, I guess, to not mess with them. They'll squirt blood out of their tear duct. So that was exciting.
So it got scared and blew blood out of its eyeballs, which sounds
Tear duct.
intriguing. Okay.
Yes.
Okay, so what are your favorite species from Australia? I assume that you probably had a connection with nature and animals in Australia that led you down this path to want this career.
Yeah, it was turtles, always turtles. But doesn't everybody, I think I wanted to start out by being a marine biologist. Everyone wants to do that, right? I'm gonna work with dolphins or coral reefs.
Oh, let's do a quick test.
And then you realize
how many
there's
people
no
in
jobs.
here wanted to be a marine biologist so that they could swim with dolphins? How many of you have ever actually swam with dolphins? Ooh, that's actually more than I thought would happen. Yes, you're right. Most of us did go into this field because we wanted to swim with dolphins.
Yes.
And okay, so there's an awesome picture of Joanna, baby Joanna, with some kangaroos. So is this like a typical experience in Australia?
When Kelly was asking me for photos, I said, well, you need the one with the Aussie with the kangaroo in the bed with you, because everybody has that, but I did have that.
That is incredible.
The mother had been hit by a car, and there was a joey in the pouch. So we had her for quite a few years. So she was imprinted, I think, with our dog. They used to go everywhere together and have adventures.
And what was her name?
Rhubarb.
Rhubarb. Okay, Rhubarb the kangaroo. Awesome. So Joanna, what have you learned after 16 to 20 years working in this watershed?
It takes a village. You need to rely on, you can't be the subject matter expert in everything. You need to surround yourself, I think, with people who can help facilitate and move things forward. Being open to thinking creatively and thinking outside the box, definitely. And I would say a big change for me was coming to Valley District and then interacting with our engineering department, who are absolutely awesome, and me suggesting that, can we make some changes to this project? And the answer was yes. And having your support and the board's support to enable us to do that. Yeah, that was amazing.
I just want to see for fun survey. How many people in this room have worked either directly with Joanna at one of her agencies or indirectly through like a field project, restoration project? That's a pretty big impact, Joanna. That's your village. Are you going to go to mine?
Oh, look at baby Heather.
Oh, there's baby Heather. This was a photo. That was a rough night at the salmon cannery. That's why I have a Band-Aid on my head.
Did you have a hard time finding photos where you weren't in the pub? Because I did.
Yes, I did. So this was my start. Actually, I think there's a few photos. So I got my start in biology. I never went to college straight out of high school. And I was just trying to find myself. And through a couple of very weird experiences, which we can talk about at happy hour, I ended up deciding to change my life by riding my bicycle from Canada to Mexico was the goal. Unfortunately, I started that journey with only $79, so not surprisingly, I ran out of money at the California-Oregon border. And so my mom's solution to this problem was, you should go get a job in a cannery in Alaska, which for kids from Seattle, that's a very normal thing to do over a summer.
Wow.
And so I went to Naknek, Alaska, which is in Bristol Bay, the largest salmonid fishery in the world. That's what I looked like every day going to work. It was very cold, and I was in the what's called the egg house, where you pack salmon eggs for the sushi industry in Japan. And for some reason, through this experience, I was really became interested in fisheries science. And this was 1997. There was no salmon run that year, or it was very small, and there was all these hypotheses on why that was. And so that's what got me interested in salmon biology. So the top right is a picture of what the cannery actually looks like on the Naknek River before they go up into their headwaters and their nursery areas. And so I didn't make any money that summer because there was no salmon, but I did decide I was going to become a fisheries biologist to save the wild salmon. And so that's what started my interest, and that's actually what I went to college for. So I knew what I wanted to do when I went to college, which I didn't do until I was 27 years old. Because in the meantime, there was some adventuring, which is these other photos in Alaska. In the meantime, I decided to become a snowboard bum. So I bought my snowboard, moved to Alaska, worked at the lift operator, so I could get a free season pass, and snowboarded every day and lived in that cabin. And that Jeep Grand Wagoneer was my car, which is why I drive a Jeep Grand Wagoneer now. But that's kind of my origin story of how I got into biology. I was really passionate about, like, why did those salmon not come back to Alaska? They are supposed to swim up that very river. Like 50 million fish are supposed to go up that river into their spawning areas, and they're supposed to return every year and complete their life cycle, and for whatever reason, they weren't there. So that's how I got into it. And then for some reason...
Is that in California?
That is actually, yes, that is in the Santa Ana River. Somehow I got from trying to save the wild salmon to saving the Santa Ana sucker. So that was an interesting journey. But like Joanna said, I went where there was a paycheck.
Yeah.
So in science, unfortunately, a lot of times you don't get to be picky about which species you're going to save.
No.
This is one of my graduate master's thesis areas.
Is that something that was repeated?
No, that was
replicates of that.
That was not our sampling technique. That was actually, unfortunately, I dropped one of the fish traps and had to stick my arm in the bayou to get it, where there were alligators and very scary looking fish. But this was a great example of like just doing field work and learning the fundamentals of how do you do scientific sampling. Maybe not that picture in particular, but some other ones. This is on the Bayou Lafourche, which is where I did my master's work. We put out gill nets, and we were sampling the total population of the fish community before they were going to take out a fish barrier. And so my thesis was on that. This is actually wherever Adam Backlund is. We were just talking about this dynamic species of California, the unarmored three-spined stickleback. This is how a lot of us got our start in fish work in California. So I worked on that up in Big Bear. And again, the cool thing is I went into biology thinking I was going to save wild salmon, and this is what I worked on, which is, not exciting all the time, but it was great. Like, this was one of my jobs, was to re-dig a pond where this native fish was that you could only do by hand. So, like, one of my jobs was literally pull grass out of a pond so that we didn't harm the fish, but they had more open water. This was when I was with U.S. Fish and Wildlife Service. And to go to Joanna's point, like, it takes a village. We were working with the Fish and Wildlife Service, U.S. Forest Service, Cal Fish and Wildlife, USGS. We all worked on these projects together to just try to move them forward. There's lots of people in this room who are in this photo. This is the Santa Ana River fish salvage, I think, when the river went dry. That is actually supposed to be water beneath our feet, but that's when the Ricks plant shut down and we had to go rescue fish. So I think the point being that you just never know where your career is going to take you. And I hope that one of the things that all the people in the audience who are young in your career is that this makes you excited about the work of the Santa Ana River. Joanna and I, we're not going to be here forever, but this permit, this plan that we're implementing, it's a 50-year permit. It will likely get extended in the future because people are always going to need water. But even, importantly, the conservation will be maintained in perpetuity. So there will always be conservation and science work in the Santa Ana River because of this effort that we've put forth to what I would consider have the win-win, which is, you have the water supply projects, you have the things that we need, but we also have conservation and science, and that's really exciting to me. So Joanna, what's your watershed moment?
I had to think about that. It actually happened incrementally for me. I think that the job I mentioned with New South Wales National Parks and Wildlife Service, recognizing that land is usually disproportionately conserved, so what gets conserved is the areas where houses can't be built. So there's, specific suites of species that benefit from that and others that do not because they are not found there. So that, I think, whetted my appetite for landscape scale conservation. And then transitioning eventually to CDFW and being frustrated with being the last cog in the wheel and not being able to make any changes or, to projects that are being proposed for implementation and a Band-Aid approach to mitigation. Then moving forward and coming over to the water district and actually being able to make a difference. I feel like I do make a difference now. Before, I think it was Band-Aids.
Joanna definitely does make a difference. Let's give her a hand for that. Joanna, I feel like Joanna is inspiring us every day to do better and to go further and to try harder. And one great example of Joanna's work is that I want you to talk about a little bit is our Enhanced Recharge project. So we were able to build 20 new recharge basins to get water in the aquifer, storm flow that needs to be captured when the conditions are wet, like right now, and put it in our aquifer for future use. And this is 300 acres of earthwork in endangered mammal habitat. And I feel like only Joanna could have gotten that done. She worked so hard on it. So can you talk about what was your secret recipe for getting this project done?
My secret recipe was Mike Esker, who's one of our engineers.
Yes.
So that is, as Heather mentioned, a really large project. We had San Bernardino kangaroo rat, Santa Ana River woolly star, no spine flower within the project area, but there was a potential for that species to be there. Gnatcatcher, and then the wild card that showed up was Leesbell's vireo, so 5 listed species, all found within the impact area for the project. To get an understanding of the scale of it, the incidental take permit that we got from CDFW, the wildlife exclusionary fencing, we had to buy 7 miles of fencing. Which created a problem in and of itself that I won't talk about now. But so initially the project looked a lot different than it does today. We went through 6 revisions, and thankfully the engineering team, after I think the initial, Mike, can you sit down while I tell you this? And then him going away and being creative enough to make changes, we reduced so the goal was, my goal at least, was to not impact more than 2 animals, 2 San Bernardino kangaroo rats. And that's what we did, but the female ended up having 2 babies. So in 300, or it was more acres than that, we relocated 2 adults and 2 juveniles. That was it. So the project changes. It was like Tetris. We'd have some biologists go out and do small mammal trapping. And then, the species are not reading the plans when it says this habitat is unsuitable for them. They're there. So they turned up in all the spots that they were not meant to turn up in. And then, so we would go out, do trapping, they'd be there, and I'd say to Mike, do you think we can lose 1/3 of this basin? Do you think we need this road here? Can we shift this a little bit in this direction? And I think that we had a really great earthwork contractor that was working with us. And after so let me back up. During the first year, we had 50 nesting birds within the project area, and we didn't stop the project. We were able to move things around, and we didn't lose any nests. So it was a huge collaborative effort between the biomonitors on the ground and the contractors. And over time, the contractors were actually suggesting changes to the project. Like, would you think if we did away with this culvert here and moved it in this direction, we'd be avoiding this really sandy area that the San Bernardino kangaroo rat likes?
Yes.
And so changes were being made on the fly as we were going along as well. And after we had put in the exclusionary fencing, we ended up catching, I think it was 3 or 4 animals along the northern boundary of the property, and I didn't want to have to move those as well. So I asked if we could shift the project to the south. We're under construction, and they did. So we were able to let those animals go at the point of capture. So all the changes that we made, again, just 4 individuals that removed 2 adults, 2 juveniles. We reduced permanent impacts from the project by 30%, and we still maintained operational capacity of the project. So that was a huge collaborative effort that I don't think I would have experienced had I not worked for Valley District.
I just want to reiterate what Joanna said and got accomplished. She turned our earthwork contractor into a conservation biologist by the end of this project. So it was extraordinary. Our engineering team and our contractor team and Joanna came up with an idea for a San Bernardino kangaroo rat bridge that we didn't want to isolate the population into 2 segments that they couldn't get across a concrete laid channel. So they came up with an idea of using an old, I think it's like the bottom of a railroad car, put it across the concrete channel, put sand on top of it so that wildlife can get across that. And I'm pretty sure that none of those contractors really knew what they were getting into when they first signed on for this project. But like Joanna said, by the end, they were suggesting how we could limit or reduce impacts of this project to whether it was nesting birds. And I also want to clarify, when she said we moved it around, we moved the construction around, not the bird nest.
Right.
So again, that's something that normally, if you tried to do that with a contractor, they would come back to the funding agency and say, well, I need this gigantic change order. They didn't do that because we established this relationship between Joanna, her team, the contracting team, all the workers out there, that we would find solutions to the problems, that we could reduce impacts, we could keep the construction moving, and we could also stay on budget. And I'm happy to report that this project got done ahead of schedule and under budget, which brings a tremendous amount of value to both the water side of the work that we do and also the conservation side of what we do. And I think it was because what I like to say is the fierce resolve of Joanna and her team and our engineering team, that they could work together to solve many, many problems every single day. So I know there's a lot of consultants in the room. There's a lot of people who are working on projects, and I just want to say, it is possible to do these things, and you do it through authentic and truthful and realistic communication with your partners on the project. Okay, I think that they're saying we need 10 minutes of Q&A, and yes, you can go back to some of these pictures and ask specific questions or relive a memory if you're in any of them. But I just want to end with, it is our honor to be in this audience today, Joanna and I, and all of the people that we've been working with at San Bernardino Valley, our board. We're so grateful for all of you in our community, my tribe, as I call it, because you guys are pushing all of these goals forward. And it does take decades. And so I'm happy to see all of our old friends, but I'm also really happy to see the new faces, because you guys are our future, and we all are here to support you going forward. So with that, should we turn it over to Q&A?
Yep.
Okay. Do you need this mic? I don't know if it's working. Any questions? Yes, sir.
We have an immigrant in our midst.
But two, by the sounds of things.
We call ICE. At what point did you become an American?
I don't know. I don't think that's a topic for the Santa Ana River Science Symposium. I only know how, Joanna, how did you become an American citizen?
I applied for citizenship after having a green card for a very long time.
What year did you become an American citizen?
Maybe it was 10 years ago.
I'll be honest, every time Joanna goes back on vacation to Australia or some other really awesome place in the Southern Hemisphere, I get scared that she's not going to come back. So we always say, Joanna, don't go anywhere really, really awesome because we want you to come back to California.
I love hiking, so I do go. I've got a trip planned for New Zealand.
See, that's what I thought she was going to say. She goes down to Australia and New Zealand and does awesome things, but she always comes back so far.
I have a more serious question. You have worked with many different plans, some of which are HCP and some of which are not. I thought the key idea of an HCP was that it was a deal with the devil. You identify the things that you want to save, and then you say, you can have the rest. And I wondered how you evaluated that, the HCP system, with the, and what happens when they overlap? They were mooching a San Bernardino HCP, which I don't think ever actually materialized, but and one of your HCPs was quite small, and the Western Riverside was quite huge. So if you could tell me more about when you do an HCP and when you don't, and what the advantages are.
Well, I'll tell you about this. So this plan is very different than most other countywide plans, like the Western Riverside Plan, which is a plan where you say, this is where our high priority conservation areas are, and everything else is kind of open to future development or whatever. Our plan is different. We identified very specific activities that are going to be constructed over the next 50 years in specific locations. We also identified the specific conservation that is used to offset the impacts of those. So it's not a typical plan where if you're in the plan area and you're not in a conservation area, you can build whatever you want. That's a totally different type of plan. But Joanna can talk about how did you analyze the impacts of these 100 different water projects. And like you said, it's not like a free get out of jail free card, you can do whatever you want. How are you going to analyze the projects going forward and their potential, which could be in 25 years, 30 years, 40 years? How have you set it up so that you can deal with those kinds of uncertainties?
So you can have HCPs that overlap. There are multiple HCPs that overlap. And Heather's right. So we do know generally where all our projects are, and we have a good idea where we're putting the mitigation, and we've already purchased a lot of the land that we need ahead of time. So in terms of analysis, at the very beginning with, estimating what our maximum impacts are going to be, because we need a number at the beginning for the plan to get approved so that we don't go beyond that number unless we get an amendment. So there was multiple levels of analysis that were completed. Most HCPs are based on projects that are ground disturbing only, so they're a little bit more easy to quantify. We have 11 water agencies that are partnered in this plan, so we have hydrology impacts as well, which is much harder to analyze, and especially when you start considering what's the interaction between surface hydrology and shallow groundwater and the groundwater-dependent ecosystems that are dependent on that hydrology, and how does this all interact. And in the upper Santa Ana River watershed, we have 5 different groundwater basins that needed to be stitched together. So we created a lot, the impacts are based on a lot of different models that were developed. So species distribution modeling, surface hydrology modeling, shallow groundwater modeling. So we've set it up where we have for each species, there's a maximum allocated amount of impacts, and there's corresponding mitigation to offset all of those impacts. And as the plan is implemented over time, so as I mentioned before, this is not carte blanche or off to the races, go do your project. Each project will need to come in when they have project-specific design plans available. We'll analyze it, look at what we identified those impacts being for that project at plan approval, make sure it's consistent, and then we'll be continuously, like it'll be like a bank account. We're going to be keeping track of things, keeping track of the impacts over time, making sure that our mitigation is staying ahead and we still have enough for the next project that's going to be coming online. And then for the hydrology impacts, all of the impacts in the HCP, as I mentioned, are based on modelling. And models are only as good as the data that goes in and the assumptions. So we will be rerunning those, updating those at a minimum every 5 years, so that we'll be able to pull in new data that's available and see, were our assumptions right in the model, and are we seeing more impacts than we thought, less? And then are there any changes that need to be made? So we'll be tracking as we go forward to make sure that we are staying within the limits of the incidental take permit.
Mm-hmm. Other questions or comments, since lots of you know us?
So my question isn't well formulated, but I'm sitting here wondering, what type of risk does the HCP or the organization, all the money and funding and stuff that you've been putting in to projects for a plan that isn't complete yet?
Yes, so that is a good question. Fortunately, we have, it's kind of funny. So even though our plan's not, we don't have our permit, our Section 10 permit yet, Joanna has been able to do separate Section 7 consultations to get permits to build the projects. So we have basically been able to construct, I think, 3 or 4 projects that are under the umbrella of the plan and also obligate the corresponding required conservation lands. How we've set it up for the risk of our investment in restoration and conservation is that if all failed, which, Brian would never let that happen, right, Brian? If the whole thing fell apart, and let's say we didn't get our permit, well, all of those projects are going to have to get they're going to have to get built anyways, right? Because they're water supply projects. And so we've set it up so that we have already dedicated, or we've already secured the conservation land that we need to offset the impacts of those projects. So we would shift in a way that either we would have to one by one go project by project to get Section 7 consultations and get permits for those, using that same conservation land that we've already set aside for the overall structure of the habitat conservation plan. And if some of those projects didn't get built, and even on the 50-year HCP, maybe not all of those projects will get built. And so Joanna has set up basically a mitigation credit structure that if we do build the projects, we will apply the credits needed to each individual project out of our, like Joanna said, kind of like a bank account. If we don't need those, then we will sell that mitigation value, those credits, on the open market to other people who need it, and that's how we will recoup the investments that we've made.
And I'd say even for those, if the individual projects moving forward, they can still rely on all of the analysis that's happened. I mean, the State Water Resources Control Board wanted to see a watershed-wide analysis of potential wastewater change petitions, which is what led to a lot of the hydrology modelling that was completed. So they're going to want to see, that analysis needs to be submitted with those petition applications. And they also wanted to see, and I would say that the US Fish and Wildlife Service and CDFW as well would prefer seeing a landscape scale mitigation strategy than, piecemeal that is not comprehensive and accounting for different species and habitats that are likely to be impacted from those projects.
Yeah. Any other questions from the audience? I see, Wendy Katagi's back there. I can see you from afar. Think of your business.
wondered if you could speak to the comprehensive adaptive management and monitoring plan and how that's looking in terms of usage for your projects as you're moving forward.
You want to explain what the camp is?
Sure. Every HCP and any mitigation area needs a long-term management plan. And for an area of 863,000 acres, 22 species, multiple different habitat types, we needed a choose-your-own-adventure long-term management plan that would guide different monitoring that would occur. And we're not going to be collecting data for the sake of collecting data, which is really frustrating. We're going to be collecting data that will be feeding into an adaptive management strategy, so it'll be a continuous loop over time. So the CAMP is the Comprehensive Adaptive Management and Monitoring Plan that was put together to cover the species and habitats that are covered by the HCP. It will be updated over time. It's going to be a living document as we move forward, so methodologies change over time. I think they used to use a narcotic to relax different soft corals when they were preserved. You can't do that anymore. I'm being told to stop.
Let her
But
finish
anyway,
her
we have a
sentence,
large
Jose.
plan that we'll be implementing.
Okay, he's telling us to stop, but I think the audience will grace us one more fun question for Joanna to end on. What was your best day in the field ever in the Santa Ana River watershed?
Oh, good grief. There's been a lot. I think seeing San Bernardino kangaroo rats. Yeah.
Well, I just want to say a big thank you to Joanna for all the hard work that she does, and also for just being part of our amazing conservation community. And we are so grateful to have you. And Joanna, do you have any last words for our audience?
Don't give up for your students. I say hassle people. It's okay to be annoying repeatedly. Go. Do you have any work this week? Sometimes it pays off. Be willing to do, whatever it takes. Sometimes jobs are not glamorous, especially for field work. Mosquitoes, deer flies, mud, poison oak, poison ivy, etc. And don't give up. I think, as I mentioned, it does take a community to move a landscape scale effort forward. And I think everybody contributes to that effort, and you have an important role to play if you're willing to put the time and effort in and be part of the system.
Yes, it's an honor to be part of something that's amazing and awesome and not common. So thank you all. Thank you, Joanna.
Thank you.
Let's give it up for Joanna and Heather. Thank you so much. That was so awesome, and I hear from them all the time. I never ever get tired of hearing stories from them, and not just because Heather's my boss, but that they have very interesting stories. So nobody asked, but my favorite field day would actually be my only field day. Two things. One, it was sad because I had to ask Kai on week 3 of working here, why does the non-native bucket not have any water in it? That is now not called the non-native bucket. It is now called the sad bucket. Number 2 is I told Kai, hey, thanks for letting me borrow your waders. I think there's a hole because there's a lot of water in here. And he goes, no, that's just your sweat. I was like, okay. This is a new adventure for me. Anyways, thank you again, Heather and Joanna. That was incredible. And now we're going to, yes, one more time for them. And we're going to continue. We have a few more speed talks and then another break. So I'm going to invite Jill down for our block 3, which is HCP in action.
Thank you so much. Thank you so much, Heather and Joanna, for that inspiring conversation. I don't know how I'm going to follow that, but I'm very pleased to be telling you about some eDNA approaches that we can be used to the end of supporting our habitat conservation plan. And speaking of it taking a village, I'd also like to take a minute to acknowledge my collaborators at San Bernardino Valley Municipal Water District, and also the colleagues at SQUIRP on this project, Drs. Susie Thoreau, Kylie Langlois, and Ryan Gilmet. So just a little refresher on eDNA in the environment, just in case nobody's used it before or this is the first time you're hearing about it, and how we're able to use it to generate some biological data like we would otherwise go out in the field and do visual surveys or electrofishing. I like to think of eDNA as the application of actually using forensic tools to answer the scientific questions about aquatic habitats that we're after. But instead of looking for who's at a crime scene, we're taking a snapshot and trying to reconstruct the biological community of who was there. And depending on the application, eDNA can give us answers to questions such as what species are present in certain different assemblages like algae, invertebrates, and fish, or what specific organisms are there and how many gene copies are present. And a typical workflow is shown here, and it involves collecting a sample, usually in our case water samples, and somehow concentrating that sample down by filtration or some other mechanism. Extracting that DNA, so pulling out all the cellular debris and other things that we don't want and just honing in right on the eDNA, and then using some suite of molecular and bioinformatic techniques to get the information out of that DNA that we want. And there are a number of molecular methods that can complement our traditional methods at the level of either looking at single species or full communities. And I approach this as just kind of a general ecologist, and my general questions are about the identification of species or the quantification of abundance in those species. And although we can't really speak directly to abundance, we can kind of roughly use these tools to kind of answer both of those questions. So two general approaches that are pretty common that we use a lot of the time at SQUIRP and in this particular set of applications. The first one is DNA metabarcoding, which resolves species richness and also identifies different species, and we can use that to characterize more full community assemblages like fish, invertebrates, and algae. And the second general pathway is looking at quantitative PCR or digital droplet PCR, which is used to quantify just one singular species and then the number of gene copies of that single organism of interest. And that latter approach is really what I'm going to hone in on for the rest of the who knows how many minutes I have left. And that approach is particularly good for screening for things like invasive taxa and other species of management concern. So pivoting this into sort of how we are using it in the Santa Ana River watershed, SQUIRP and San Bernardino Valley have honed in on these two different DNA techniques to answer questions relating to species of interest such as the golden mussel and the Santa Ana sucker, which I will tell you about now. So the invasive golden mussel has been a concern throughout watersheds connected to the citrus reservoir. There's a map there in the bottom left. It was first detected in Stockton in October 2024, I believe, and there's now a widespread effort to test for its presence. And because of the interest and the deleterious impacts of having this particular mussel take over this watershed, there's actually a pretty robust ddPCR or digital droplet PCR assay, so a molecular tool that's used that's just hones in on that specific golden mussel species. We can use that for both detecting and quantifying the number of gene copies in the water column of that. In the citrus reservoir, we use a Smith root auto sampler deployed for over the course of 8 days, as well as some grab samples using a handheld Smith root pump on the first day and the last day, sort of around the reservoir. And then we also put out settlement plates to look for golden mussel villagers. And the good news is that as of April 2025, which now seems like kind of dated, there were no positive hits for golden mussel in citrus reservoir. But now the golden mussel has encroached further and further closer to this reservoir, and we're trying to come up with an eDNA-based routine monitoring. So the next kind of thing on the plate that we'll be tackling next with our community of practice is a statewide lab intercalibration to make sure that all the labs that are testing for golden mussel are able to do so at sort of the same levels of certainty. And I believe our samples are actually in the mail right now. They're going to land in our office on Monday, which is pretty exciting. So the second eDNA test case that I like to tell you about is about our friend, the Santa Ana sucker. The Santa Ana sucker, as you all know, is a freshwater fish that uses the Santa Ana, San Gabriel, and apparently the Santa Clara River. I learned that this morning. And it's been threatened since about the 1980s due to habitat loss and hydrological modification. And this species, among all the other ones, are surveyed annually through the annual fish survey using electrofishing, which I took part in this past year for the first time, which, to my surprise, involves briefly stunning fish in their habitat, IDing them, weighing them, and collecting measurements, which I found to be very fun, but potentially invasive, to the fish itself. So per the Habitat Conservation Plan, efforts to conserve and protect this species do require ongoing monitoring, which is best done non-invasively in a complementary way. So eDNA is looking to fill that gap, sort of, between those annual fish surveys. So we actually have a whole bunch of water samples that potentially have Santa Ana sucker in them that we'd like to test. But we're actually starting in square one because unlike the golden mussel, the Santa Ana sucker did not have a reliable quantitative assay developed yet. So SQUIRP stepped in and developed an assay in silico. So on the computer, we developed this assay that's specific to just the Santa Ana sucker. And we actually finalized it last week, which is kind of exciting. So the next step is to validate it using actual tissue and then further validate it using field samples that we had taken through the San Bernardino Valley team. And one important benefit to using this molecular tool is now we can no direct handling of that species is actually necessary other than how we monitor it generally. So future work also involves developing similar assay. I've got our wish list species here. You probably know what those are, the speckled dace and the arroyo chub. So they're next on deck if this works out. And for all the nerds in the room, in case you're wondering how we do that from scratch, in order to create a novel primer for the Santa Ana sucker, we downloaded reference sequences from GenBank and a few other sources and got as many reliable sequences as we could for a total of 55 sequences covering a particular region of the genome. So the next step is to line them all up so they're all in the same place, that covering the same region of the gene. And for this, we selected the ND subunit 2 part of the mitochondrial genome of the Santa Ana sucker. So we lined them up and then we honed in on a smaller target region that is conserved across the individuals. So those 55 sequences look the same across individuals. And then we compared those to similar species, so other suckers, and looked for differences so that we could distinguish between what was the species and what was maybe a closely related species, since we don't want to be detecting other suckers. And then we also pulled from the annual fish survey some other sympatric species that are found in the river. Took those sequences to make sure there wasn't any overlap between things like channel cats, western mosquito fish, and fathead minnows. So after we had something suitable, we checked it by running in silico PCR, so basically on the computer, simulating what a PCR reaction would do, and then seeing what kind of amplicons we were getting and what species those were hitting. So after that, it was sort of a, chemistry checks to make sure everything checks out. And so these steps were iterated 19 times until we got a promising assay. So just to wrap this up, here we identify a couple of use cases for eDNA tools and management to non-invasively answer questions about species of management concern. The first was screening for the golden mussel, which seems well in hand, and then also for the Santa Ana sucker and other species of management concern. So I think I have a moment for questions, but thank you. There's one in the back.
I hope this is worth it. That was a big production to get the microphone to me. So I'm curious, with the methods right now, what is the spatial and temporal scale of resolution? Like if you go out to a location and you sample, and you get a positive read for Santa Ana sucker, so we think Santa Ana sucker is here. What is the spatial scale of here that you sort of feel comfortable assigning?
That's a really great question. It's also a very
Thank
open
you.
question in terms of the eDNA community. Is sort of, I like to think of it as, it's a fingerprint. How long does that fingerprint realistically stay for in an aquatic habitat? Things like that. What we do know is that in an aquatic habitat, the eDNA signal persists for about a day if it's in non-moving conditions, and there's been a few tests done to see, if you put something in and then pull it out, how long does that signal persist? It's usually gone in a few minutes. So I'd say we're pretty certain that there is a Santa Ana sucker there recently, but I can't exactly say, how many or for how long. Great question. Okay, I'm gonna turn it over.
All right, well, yeah, thanks for having me. So today I want to talk a little bit about some preliminary analyses I've been doing, trying to develop a stock recruitment relationship for Santa Ana sucker. So none of this would be possible without RCD and Valley District especially. I just have a few of my co-authors listed up here. I have no good pictures of Kai or Kerwin. They're always taking the pictures, not actually in the pictures, so these are the best I got. I got to get some pictures from you guys. And then you've already seen the one of Brett with the car in the middle of the river. So I love that picture. It's always in my presentations. So Santa Ana sucker, federally listed species. So obviously there's a recovery strategy or a recovery plan. And so in that recovery plan, when reading it, you notice that it emphasizes, or at least it stood out to me, that there's no PVA that's ever been really developed for Santa Ana sucker. And this is kind of a concern because a PVA is a really useful tool for management, right? So a population viability analysis, if you want to understand the threats to persistence or how some kind of action might affect persistence in a threatened species, a PVA tool is really useful for assessing that. That being said, the recovery plan was actually written or published in 2017, I believe. And so we've actually recently put out a PVA using our adult surveys. So the native fish surveys that you've heard talked about today, we've actually put out a paper in 2024 developing a PVA from that time series accounts. However, even though this is, I feel good about this PVA, it's really limited in a number of ways. And one of the big ways is the PVA, it's only built on the adult population, right? It implies mechanisms affecting the juveniles or the recruitment process, but it doesn't actually capture the recruitment process. And the reason this is a concern is because Santa Ana sucker are these opportunistic species, right? They have life history characteristics that suggest they have early maturity, they grow really quickly, they have a very short lifespan, and they're pretty fecund for their size. So kind of in between that opportunistic and periodic life history trilateral continuum. And so why is this important is fish species that usually demonstrate these life history attributes typically their population growth is really affected by mechanisms operating on those early life stages. So the recruitment process and survival during those early life stages, that typically really controls your population growth rates. So it logically makes sense that we'd want to understand that recruitment process. And so even with basic count information or time series in our surveys, oh man, that equation really fell apart. Don't worry about the equation. But essentially with these, we can build these stock recruitment relationships, right? So we have tools. We have the Ricker population growth model for people who do any kind of fisheries work, probably familiar with it. But there's ways to build these relationships between the stock, those individuals reproducing, and the recruits produced. We can build these relationships and assess different mechanisms that might impact that relationship. And so that was essentially the goal of this preliminary effort is, can we actually establish the stock recruitment relationship? Can we actually identify mechanisms that might impact that stock recruitment relationship? And then ultimately, can we actually use this stock recruitment relationship to project the population into the future? So essentially build a PVA. And so The native fish survey, this is essentially our source of stock data. So again, a lot of people who have been in here have been out helping us do this survey in the summer. Essentially just electrofishing, trying to get an estimate of population size of the adult population. So again, this is occurring in the middle Santa Ana River, about 16 river kilometers, essentially is where this modeling is taking place. In addition to that, Valley District and the RCD do annual larval surveys as well, and Kai actually mentioned this during his talk, but essentially just walking the stream and actually counting those larval fish at repeat sampling locations. And so this essentially gets our recruitment information, right? And so what's nice about this is that's really all we need to build a stock recruitment relationship. The longer that time series, the better the model works. In addition to building that stock recruitment relationship, we can also build some flexibility into that model. So what kind of environmental conditions might actually impact that stock recruitment relationship? And so what we did is we built in a temperature effect, a discharge effect, and a substrate effect. And so the temperature effect, go to my poster this evening if you can, and I'll talk all about the temperature model that we actually built. But ultimately, we quantified a metric that was essentially represents how many days we're within that spawning window every year. And so obviously, we would expect that to affect the recruitment process. Discharge, we used a spring discharge variability metric. This is an opportunistic species. We expect reproduction to occur when the conditions are opportunistic, right? So very flashy systems, that's what they've evolved to. So if there's a lot of variability in the flow regime that spring, it's conditions that are really suitable for Santa Ana sucker spawning. And then lastly, Santa Ana sucker in the Santa Ana River are one of the most common hypotheses you will usually come across is that they're limited in substrate, right? So there's not a lot of good spawning substrate along the river. So gravel cobble is what they need. So it logically makes sense to try to model that recruitment process as a function of those covariates. And so essentially I fit this model using Bayesian methods. So each one of these lines represents a posterior distribution. Happy to talk about the details if you want. But ultimately, this creates that essentially time series, right? So we have our stock and then the recruits that are produced from that stock. And so from this information, we not only can look at the environmental factors that affect that stock recruitment relationship, but ultimately we can build a PVA. Again, this is very much preliminary. But ultimately, I just want to show this figure to demonstrate how these can be valuable. And so the idea here is we can estimate by projecting that population into the future, giving those conditions, that stock recruitment relationship, and estimate what's the probability that the population would go extirpated under these different conditions. And again, I just use that variability in spring flow as the metric here. But as you can see, just based on these simulations, if we have those kind of average conditions in terms of how much variability there is during that spawning window, that seems to be the best conditions. We have our lowest extirpation risk. Again, very much preliminary, but the value of this tool, I think, is really what I want to communicate. A lot of things that I can discuss right here. I'm not going to for the sake of time. But I do want to emphasize that, again, this is very much preliminary, and there's a lot of complexity that really gets ignored from this model. And so this is just kind of a basic life cycle model I've kind of schemed out, trying to get an idea of what I can do to actually improve this model. And so in this example, this is just showing a very basic, the stock recruitment relationship, right? We have our stock as the adults, we have our recruits as the larvae, very simplistic relationship. But there's obviously a lot more complexity that can be built into it and should be built into it to really get an understanding of what really limits the population. Too many people to thank, really. As you saw, the majority of people in here raised their hands when we talked about who went to the native fish surveys over the past year. So all of you, absolutely important to make this happen. And then funding sources, Valley District, supporting a lot of this research. And then RCD, they always provide a lot of the bodies and really make this possible. So this is just some citations if you're interested. I can also bring this back up if you're interested later. We also have the native fish survey is actually publicly available. I don't know if this actually link works anymore, but it should. If it doesn't, all of our data is published on ScienceBase, but from what I was told that our ScienceBase page, at least for all the data, was decommissioned, and so now you have to go for each individual year if you're looking for the data. So if it doesn't work, I apologize, but it should. And so that's everything I have. No questions? Well, I don't have any questions either.
Thanks, Brock. That was super interesting. I'm curious of what you think for future applications of the modeling. Like, are you interested in looking at future flow scenarios or climate scenarios? So what's your take on that?
Yeah. So I have a lot of interest. So I'd love to build in like the non-native fish. So there's an additional complexity that's not shown in this just because this is a speed talk. But being able to make the recruits and the stock kind of communicate and be on equal scale is kind of an involved process. And the same thing would happen with bringing in the non-natives. So obviously, we would expect the non-natives to also affect the recruitment process. The non-native fish are actually collected during our non-native fish or our native fish surveys as well, and making that actually at a comparable scale creates some complexity. I've actually done it. It just I don't like the way it looks, so I didn't use that model presenting here. But things like that, anything that might affect that recruitment process, finer scale discharge kind of information would be really great, especially given the wastewater treatment facilities upstream, and being able to say manipulate that and see how that might impact persistence would be really cool. Right now I'm just using the USGS gauge at the MWD crossing, so it doesn't reflect that fine scale variability in discharge. All right, cool.
Thank you.
Hi, everyone. I'm Chris Taniguchi-Kwan. I lead the Eco-Hydrology Research Program at Southern California Coastal Water Research Project, as Rafi said, SCORP. And really excited to be here today, and I feel super inspired to hear about all the cool work that's going on, all the data that's being collected, and all the coordination and monitoring efforts that are going on in this watershed. So today I'm going to be talking about a new project that's just being initiated and kicked off in partnership and funded by Orange County Water District. And it's all about balancing water use and habitat protection for the Santa Ana River at Prado Basin. And I was super happy to hear Nate's talk. He really set up kind of the framework. So for this project, we're also using the California Environmental Flows Framework, and he applied it in the LA River. And for this case, we're gonna be taking a look at the Santa Ana River watershed. So super excited. As many of you know, Prado Basin supports multiple different benefits from flood control, groundwater recharge downstream, critical riparian habitat. This is an aerial view of Prado Basin. Oh, oops. I don't know how to get to the pointer, but the dark green area, you can see the dense riparian forest, really important for the leaf spells vireo and other riparian habitat. And then you see inflows from the Santa Ana River that come in the inlet of the basin, and then you have Prado Dam at the outlet that really supports flood control and groundwater recharge downstream. And so we know that in the future, stormwater capture, both wet weather and dry weather, and the increase in water use are all gonna change inflows into Prado Basin and ultimately change the stream flow conditions within the Santa Ana River and the major tributaries. And so one of the key questions that we have is how will these changes affect aquatic and riparian habitat across the watershed? And so ultimately, managers need science-based tools to help support balanced decisions that can help maintain both water supply reliability and also protect ecosystem health. And so this project is all about developing the tools to help inform these types of decisions. And as many of you know, Prado Basin is the focal point for this, but actions and decisions made at Prado Dam are really connected to a broad set of watershed actions in the watershed, from reservoir operations at Seven Oaks Dam, stormwater capture, increases in water reuse. All of these conditions are connected. And so one of the key questions that we have are, what are the ranges of stream flow conditions across wet season and across the spring and the dry season and the fall that are needed to support sensitive species that we're trying to maintain and protect in this watershed? And so the major goal of this project is to help develop tools that can help inform those ranges of flows that are ecologically protective. And so these are our key questions. The first one, as I mentioned, is all about understanding the hydrologic conditions within the middle Santa Ana River and some of the major tributaries needed to support both riparian habitat and important aquatic habitat for Santa Ana sucker and some other aquatic species. And then the next phase of the project, the second phase, is all about thinking about future changes in inflows and reservoir operations and how that might impact stream flow and ecosystem health. And that's kind of why I was asking the question to Brock of, oh, can you use that model, look at those future conditions and scenarios? And it'd be great to chat more about it after this. And so for this project, we really aren't collecting a lot of new data. There's been a wealth of data, information studies that are going on and existing tools that we can try to leverage. One of the tools that we're using is the California Environmental Flows Framework. So this is a science-based framework that provides managers guidance on how do you actually determine environmental flows that seek to balance water for the environment and water for human uses. And so there's a scientific process, there's a set of statewide tools and different resources that are readily available. And so we're kind of leveraging this framework as a starting point. And so ultimately we're going to be applying CEF, and there's 3 key components to this study. The first one is understanding what are the baseline hydrologic conditions. And so we're going to be evaluating kind of the natural flow conditions using existing models that are readily available on the Natural Flows database hosted by the Nature Conservancy. And then the second piece in all of this is zooming into specific locations of interest and thinking about more site-specific details about the channel morphology and about specific habitat needs for sensitive species like the Santa Ana sucker. And so we're going to be developing and updating existing flow ecology models that relate important physical habitat characteristics like depth and velocity to the needs of different species life stages, for example, the Santa Ana sucker. And so ultimately, the outcome of A and B are really ranges of flows that we think would be supportive of broad ecosystem functions at different locations across the watershed. And then section C, which I mentioned earlier is the second phase of this study, is when we start to think about future management scenarios, future stream flow conditions in the watershed, and we start to look at potential trade-offs across all those different management objectives, flood control, water supply, so on and so forth. And so this is a conceptual example of a flow ecology relationship that could be developed through this study. That this figure really shows that as you increase inflows into Prado Basin, for example, you increase conditions for suitable habitat for different endpoints. And so ultimately, we want to develop some kind of flow ecology relationship across different locations in the watershed that can really help inform the sensitivity of the system as you incrementally change flows, whether more or less, and also to look at potential trade-offs if you think about these different management objectives as well. And so as I mentioned, we're just getting started on this project, and so there's multiple different ways for any one of you to get engaged on our project. From becoming a project partner, if you're like, hey, I'm really interested in temperature, are you looking at that? And we're like, oh, I want to look at that, no. Or to participate on our advisory group. So we're going to be forming a stakeholder advisory group and a technical advisory group, and we really want to lean in and learn from everyone that's really been working for decades in this watershed. And then the last piece is data and information sharing. So we'd love to hear from you, to meet with you, to hear about your perspectives and your goals within the watershed, and try to incorporate that into our study. So with that, feel free to email me or talk to me after this.
So this is probably pretty early for you, but what focal species besides Santa Ana sucker are you thinking of looking at for the CEF?
Yeah, so right now we are the Santa Ana sucker. We're trying to build off of existing work, all the amazing surveys and physical habitat data that we have. And then also the willow riparian habitat as another endpoint. And I'm really interested to find the person who was talking about the kangaroo rat, because I was like, oh, that would be really cool to look at, like in terms of the peak flows, to bring that perspective in. And so, but that'll kind of be a part of the process too of which species of concern and do we have the data to kind of look at that. Oh yeah.
Hi, this is really great you're doing this. I was wondering, and I used to be at the Army Corps, so they care about flood flows and then a lot of water interests. So are those voices going to be integrated into the flows, scenarios, and trade-offs?
Yes, absolutely. I didn't have enough time to talk about kind of like the ultimate vision for all of this. So OC Water District has spent a lot of time with scripts developing different models and relationships for their FIRO, Forecast Informed Reservoir Operations, specifically looking at vireo habitat in Prado Basin. The ultimate goal is to kind of bring this in. So kind of taking their information at Prado Basin and integrating that into their system with the Army Corps and bring in all the different perspectives. Thanks. Oh, we have time for one more?
Thanks for this very exciting project, and just wondering if you could share a little bit more about the scenarios that for the future, because we heard the LA River this morning, which has like such a big, bold, long-term vision. How is that going to be type of thing going to be integrated here?
Yeah, so I think we haven't entirely scoped out what scenarios, so we're hoping to integrate and coordinate with the HCP and a lot of different agencies to help identify what those future scenarios are. But initially, kind of our first conversations are like building off of the planned wastewater change petitions and stormwater captures that are like being considered. And I know there's been a lot of data information in terms of like changes in discharges. So we've really only talked about the hydrology piece, and we haven't really talked about like stream restoration or all the other management actions that could be put. But yeah, for now I think it's hydrology, but I think there could be a potential to expand that.
Thanks.
Yeah, thanks.
Thank you, everyone. We're going to take another break. We've got some snacks out there, so please enjoy and then be back at 3:20. 3:20. Thank you. All right, we are back from another break. Did you have a cookie or chips? Both? Excellent, excellent choice. So we have a few more speakers, and then we have our final panel, and then we will head over to the cafe for our networking event. So I'd like to invite up Mariana for her talk.
So, hello everyone. Hello, my name is Mariana Antonio. I am a grad student at Cal State San Bernardino, and my project is From Genes to Streams, Mitochondrial Approaches to Population Monitoring of the Santa Ana Sucker. In this presentation, I will go over the Santa Ana sucker, which I'm pretty sure most of us are familiar with. And I will also go over the mitochondrial genome and environmental DNA. Then I will go over my methods, results, and future directions for this project. So to start off, the Santa Ana sucker can be found in the Santa Ana, San Gabriel, and San Clara River, as it was mentioned previously. It is federally threatened under the U.S. Endangered Species Act in the early 2000s, and since the 1970s, about 95% of its natural habitat has been lost due to degradation, competition with non-native species, and water alterations in the river systems. So we were really interested in looking at the mitochondrial genome, since there have been multiple studies that use it to create specific genetic markers that can be used in eDNA studies. So that is something that we wanted to do with the Santa Ana sucker. To introduce the mitochondrial genome, it is maternally inherited, so only one gene is passed down. It is used in population genetics as well as phylogenetics and has more mutations than nuclear DNA. So right here, I just have an example of what the mitochondrial genome looks like. It ranges from 16,000 to 17,000 base pairs and has a total of 36 genes. Now, moving on to eDNA. I know previously we had a talk. It was really nice. To introduce again, eDNA is any genetic material that sheds into the environment. So this can be air, water, soil, and our lab is mostly interested in water. So it is an exciting method to study and monitor biodiversity in an ecosystem, since some traditional ways can be costly, time-consuming, and required trained personnel. Some current ways that they are surveying for fishes has been backpack electrofishing, netting, tagging, and video software. So by implementing eDNA, this provides a method of non-invasive sampling that allows for population survey without disturbing the species, since you don't need to have direct contact. You don't need to capture the species or add any stress. It is also more cost-effective and demands less physical labor. So it is a great method if you're working with a sensitive species. Right here I have an example of what eDNA collecting could be. This is using an IV bag where we collected some water and then filtered it through this filter. Any DNA that is present in that water will get caught on. However, the current methods that we are using are this eDNA spheres. Again, it has a filter, and this one, you just put it in the water, and any DNA that's in the water will be captured in, and then we will process the DNA. Moving on to metabarcoding using eDNA. So metabarcoding is a process of using DNA barcodes to identify multiple species in one single sample. This can be done by using PCR universal primers and is my working? Yep. Oh, okay, sorry. And reference database. So right here we have the universal primers. We have region 1 and region 2, which are the same since they're universal, but we're really focused on this variation area since this will have the unique barcodes. So it will tell us, are we dealing with species A, species B, or species C? So my overall goal for this project is to sequence the Santa Ana sucker's mitochondrial genome to generate primers that will be used to detect the presence of this fish in the different river drainages. And so far, my methods were the DNA extraction. Like I mentioned, the Santa Ana sucker is sensitive, so we couldn't just simply go and get one. But fortunately enough, we were able to get some tissue samples from the Natural History Museum of Los Angeles. They were deposited by Max Murray back in 2023, and using a blood and tissue kit, we were able to process and extract the DNA. Then we did PCR. This is polymerase chain reaction. So we were able to create multiple copies of a specific DNA fragment, and then using CAP primers, which are unique to the Catostomus, we were able to sequence, we were able to get I don't know how you go back. We were just able to introduce primers to the entire mitochondrial genome. Then we wanted to make sure that these primers were actually binding. So we wanted to visualize our DNA fragments by using gel electrophoresis. And the way that that works is that the DNA is separated based on sizes using electrical current. Lastly, we did DNA sequencing using next generation sequencing. So that's PacBioReveal and Oxbow Nanopore. For the PacBioReveal, we went to UCI. So right here, this is the big machine that they use. This machine gave us circular consensus sequences that provide more accurate reads. And then for the Oxbow Nanopore, we use Plasmasaurus to give us linear PCR standard purified sequencing. So we were able to actually sequence the entire mitochondrial genome. This is very exciting because I have been working on this for 2 years. I don't know if you guys have been here in the previous years. I've been doing the poster, so slowly I was able to sequence the entire mitochondrial genome, and that gave us a total of 16,631 base pairs. And I was able to get this figure by using mito-annotator programming for mitofish. So I'm really excited about that. Now that we have the entire mitochondrial genome, we're actually able to design specific genetic markers that are unique to the Santa Ana sucker. So the way that we did it is we grabbed 2 suckers. We have the Catastomus ananidae, the sequencing that we have, and then we have the Catastomus discobolus, Catastomus bondi, and Catastomus catastomus. I used Mega 11 to align all of them, and then identified more loci sequencing to help distinguish the Santa Ana sucker and see what's different compared to these other fishes. And then once I was able to see that, we used primer blast and got 10 potential primers. Then I calculated their overall genetic distance. We wanted to look at high genetic distance since we knew that these suckers are closely related, and we wanted to make sure that we were able to distinguish the Santa Ana sucker from the other suckers. So with that, we were able to get 3 potential primers that had their overall highest genetic mean distance, and then right now we're just currently labeling them as SAS5, SAS6, and SAS7. So for SAS5 and 6, it is located in cytochrome B, which is pretty typical since there are multiple species that already use cytochrome B in different species. But for the SAS7, for that one's in the NADH. And we wanted to make sure that these primers actually worked. So with the previous extraction that I did a while ago, we still have the DNA concentration, and we tested it. So as you can see right here, our primers are binding. This band right here indicates that our primers are recognizing the Santa Ana sucker and are binding it. So that is also very exciting to know that our primers are working. Now, for future directions, we want to continue developing our eDNA protocol, as well as test our SAS primers in different environments. And currently, our eDNA protocol is, again, using the eDNA spheres, where we collect DNA. We collect these spheres, we collect eDNA using these spheres passively, so we do not get any water from the river, we do not get any water from any testing sites, or have any contact with the species. We just put them in the water, and then for 20 minutes, and then any DNA that's present will get attached to the filter. Then we process them using a different kit. This is an rRNA water kit. And then for the PCR, we use the primers that I mentioned earlier, the 3 primers. And if we see that that there is a band, then we know that the Santa Ana sucker is present. And we also, moving on to the future directions, we also want to test it in different environments. So far, we've only tested it in the 7 Elks Dam, where we found no fish DNA present. But our future locations are the Riverside Corona Resource Conservation District Facility, and then the Martha McLean Aza Narrows Park. Lastly, I want to say thank you to everybody who made this project possible, and I will open the floor to any questions. Thank you.
I kind of have the same question as the guy with the other presenter earlier. So for how long in one area, or how big of an area, if you get a positive sucker hit, is it going to be that pool that you're in? Would it be within the last 5 minutes? Like how long and how big of an area would it be accurately testing?
Yeah, like it was mentioned before, I don't really know how big of an area or how far. I do know that have been studies that suggest that DNA can be there up to 7 to 10 days. And I'm sure there's also different factors that will determine how quickly the DNA degrades. So I'm not sure about the range.
Can you do remote sensing, or is it ongoing?
Excuse.
Could you leave the sensors in the creek like permanently where you'd have remote sensing capabilities, so if the time wasn't real long, if a fish swam by, you get a hit? Or is it something you have to go out in person and collect each time?
We don't use any type of remotes. The spheres that I was talking about, we just place them in the water for 20 minutes and then take them out.
I'm just curious, I had this question actually with the last eDNA, and this might just be a bigger question. How does eDNA then I understand it generally from a presence-absence. How do we move into population monitoring? Like, is there something with the DNA sequencing or the test that gives you a sense of, did one fish go by or 20 fish go by? Or is it just, you would then have a broader sampling program across, I'll just stop guessing, but that's my question.
Yeah, that's a good question, and that's really interesting, something that I, too, was interested when I got into eDNA. I don't believe that there is any way, like, surely of knowing currently how much there is. I know it's just the concentration of where you were in that site. Okay, thank you.
Okay, thank you everybody for having me today. My name is Rachel Pickstein, and I am from Loma Linda University, where I just started year 3 of my PhD. But I'm a little bit of a unique student, so I'll share a little bit about that. First of all, I am here to present about what I have always been told to refer to it as, and what I believe it is best described as, is the nasty necessity. Invasive species are not fun to manage, they are not fun to remove, they are not fun to exterminate, but it takes somebody to do it. So when it comes into this in a watershed, I will tell you I am a terrestrial ecologist with a background in environmental ecology and conservation. My specialty is herpetology, particularly saurology, or big lizards and big snakes. And I also have worked with venomous for over 20 years, which is why I'm in Bill Hayes' lab, Dr. Bill Hayes at Loma Linda. And we do behavior in rattlesnakes, birds, venom complexity, and behavioral ecology, with also a little bit of work in venom complexity with invertebrates as well as fish. So I had to learn about the watershed, and I thank you for bringing me here because science is about consensus. And so a little bit about myself. I'm originally from Michigan. I currently live in Arizona, and I actually commute weekly to California, that 5-hour trip to the desert, and live between two places. Between going from being a teacher and college professor from grade school to undergrad, I was at GCU for about 7 years, but I taught for about 15 before going back for my PhD 2 years ago. In that transition, I started my own nonprofit, which is Pickstein Laboratory for Conservation, Research, and Education. Pickstein Lab's goal is, through public outreach like Comic-Cons and different fundraising programs, going to reptile expos, big on advocating for better captive, or really the best, setting the standard, raising it for captive husbandry, connecting zoos to hobbyists, to research, to academic, to improve herpetology and exotic pet ownership as a whole, because they're kind of the start and the end of what I do, because I specialize in, as you could tell from my logo there, tegu lizards. And I fell in love with them first as a pet owner, learned their plight, and became dedicated for life to these little guys. Well, pretty large lizards, if in lizard stance, really. So because of my awesomeness of my co-authors from Texas Christian University, Dr. Dean Williams, to my PI, Dr. Bill Hayes, who agreed to have me bring this project after 9 years of it being self-funded to Loma Linda to continue it as a PhD, I focus on the genetic ecology of invasive tegu lizards in southern Florida, and looking at how things like phenotypic plasticity makes them more formidable. But one of the many things, whether it's like disease and fungal infections, all these things are not really looked at a lot from exotic species exposure, and it's a whole aspect of invasive species we miss. And so I'm big on looking at, as these articles build, what the real story of this species is. So they are the largest member of genus Salvator of the Tiid family, which are our whiptails. You have a lot of them in California. We have nearly over 100, I believe, in Arizona, and includes the amoebas as well. And they are, of course, black and white and band all over. over. They now have legislation in about 6 states, and it's unfortunate because it all comes from the pet trade, but they do go back about 50 years in the leather trade. And they once were, about a decade ago, the world's most commercially exploited reptile. So you have them in one stance being commercially exploited in their native home, and then also imported for the pet trade, removed as an invasive, studied as a lab rat, long-term. A lot of vertebrate studies, spermatozoa, liver functions, all this great stuff. But who's studying the animal, their behavior, their ecology, as it dwindles? They are convergently related to the varanid family. There are a lot of differences when you see beyond the skin deep. And in general, like an invasive species, they are a problem in every manner. And so I'm very, very nitpicky on the fact that we use this word correctly. A lot of people, for example, put millions of dollars, I couldn't even name on 2 hands how many research labs studied the Mediterranean gecko in the United States, and they have only hypothesized disease spread. I understand we can't just let that go loose, but we have animals that are demonstrating massive amounts in every single category already, and we're not putting allocated funding towards it. We are not doing anything preventative in the United States with invasive animal removal, minimal with plant removal, and it still has a lot of political barriers and ridiculous things that get in the way of solutions. The thing I specialized in my master's was invasive species from cooperative collaboration, looking at what it takes to solve it. When I graduated in 2016, there were less than a dozen complete eradications of invasive species of any kind removed globally. Today, there are less than 25. So not much has changed, and so we overlabel things invasive, while we have other things that are non-native established that, are not, going to get as much of the funding. So it's improperly over adding an invasive term, like the green iguana in Florida, arguably economic damage and not ecological. So we get on these little red lines of tape and we get hung up. And I like to point out, for example, that the Rapid Response Tegu Management Plan, that was the name of it in South Florida, took 2 years itself to release, while over 4,000 minimum estimate animals were released as hatchlings with 90% survivor hatch rates and arguably only a 10% mortality rate at its highest. These animals reproduce fast, and we are not responding quick enough. So when we look at an invasion curve, when public awareness becomes happening, we are now at a point where we are not able to remove them. And so the media and the politics, all this creates this horrible triangle that we are not able to address. And what it takes to solve invasive species problems is not only being incredibly preventative and ahead and predictory, and we have the science, we have all this ready, we just need to implement it. And so it takes bringing, arguably, other countries, states, organizations, different scientists together. The key to solving invasive species is collaboration, both the preventative and the reactive side. But sorry, Florida, I don't think it's in your budget coming up anytime soon. They give about $1.5 million towards invasive plants every year. There is still no budget yearly for animals. So let's talk about the Santa Ana River watershed. Interesting to look it up because we've got a lot of things that were new to me and a lot of things that were not. So when we look at our aquatic species, we have different clams. We have some different species of that plant that live at and near the water. We have the American bullfrog eating everything. We have the silver carp doing what we see in the Great Lakes, right over here behind me where I'm from, polluting everything. Red-eared slider, the lost pet story, old, tale as old as time, and a lot of interesting plants doing, typical invasive plant stuff. So first of all, you guys already know, this is what I learned about you. You're a major Southern California watershed. You have critical riparian and aquatic habitats that are integrated. And you have an incredible supporting of endangered and sensitive native species, arguably some of the most endemic species in the country that are not in the Florida through Mississippi region is in California. And so we have incredible species of amphibian that are at risk, all sorts of little cuties that I really enjoyed looking up, and all sorts of lizards, including those in the Tiid family, including a specific horn lizard. There's a beautiful whiptail in the center bottom. Aren't they a little tegu? Look at that face. That's a little tegu. And people who study whiptails, by the way, call them big whiptails. You guys also have some incredibly unique and varied species of snake. And so there's a lot to protect here. We have habitat degradation, alteration, competition with native species, threats to endangered fauna, nutria, never a good rodent to have. And a lot of these things, if we look at them and go back individually, it's a lot of waiting too long to react, and then it just creeps in. Now, the good news is a lot of these things are not in the watershed and river directly. A lot of these things, when we look at the maps, are on the edges, like our African cloud frog and bullfrog, are not permeated through the entire system. But when it comes to it, there is basically an invasive species up against every aspect of the watershed, whether it's at its borders or already within it. And every one of these are going to cause different complications and issues. So the best advice I always give is prevention. But the most important thing to do from this point, whether it was day 1 of the introduction or day 10, sorry, I ran out of time, so I'm just clicking through my slides here. Because you guys all know what this stuff is. I'm preaching to the choir here. But this is what I learned going through this. It's about collaboration. So having a person like me who knows nothing about aquatics really, and has just learned about your watershed in the last 2 months, showing up to talk here, this is why we're here. It's about collaboration, because I might have people or resources or tools, or I might have gone through something that allows you to apply to the programs you already have into existence, to add to new ideas, because science is about consensus. And when it comes to invasive species research, we have to, of course, move beyond just the government agencies, the public awareness, and the media, as well as the scientists in the front, and take ideas from all sectors, because it takes all of us working together to go beyond the issue. And of course, we are 80% of the cause of the problem globally. Thank you. Do I have time for questions? And I have time for questions. I can go into specifics more on anything. This was meant to be more of a overview. But in general, yeah, I'll take if there's any questions at all. Okay, so I would be happy to talk a little more of my tegu stuff. One of the most interesting things I found is that there is hybridization between Salvatore rufescens, the red tegu, and Salvatore marinae, the black and white tegu, which are the 2 largest members of this genus. There's 3, but 1 has never been actually imported. And we were able to discover there was hybridization predicted in 2017, confirmed in 2020. I've been able to confirm in my samples that they are coming over with this rufescens DNA in them, already hybridized. And it's actually adding to the phenotypic plasticity, making them more adaptive. Keep in mind, tegu lizards like Salvatore marinae have already been shown to be able to adjust their body temperature seasonally during breeding seasons and have endothermic capabilities, similar to what we see in great white sharks. And so if you already have an animal that's already phenotypic plastic, and you find out it's even more so, it's just going to spell a more formidable animal, which is why it was able to check so many boxes in basically every category of being an invasive species. And so I would recommend not only do we collaborate, but we need to look at genetics. eDNA is fabulous. It's like really remarkable. They're doing it from the air now too, with fecal from animals and forest dumps like okapis, and understanding their entire diet structure. So really collaborating with techniques and tools will allow us to better understand it. But I can't emphasize enough the importance of understanding genetics to really understanding how formidable the invasive species is within the system, not just its body count. So, yes. What is the method for control? For my lizards? Oh, yes, thank you. So when it goes to most vertebrate or invertebrate, it's about removal. So it's about humane monitoring and euthanasia. So for these lizards, there are traps. They are basically raccoon steel drop-style traps that you could buy from Home Depot. They are often just left bright steel on the side of the road. That's what Fish and Wildlife does, unfortunately. Some of my training went in there, and we tried to get the money in the budget to basically say, hey, we have to spray paint them green. We have to be careful if it doesn't become too strong of a smell that it deters them. We have to cover them with plants. We usually wrap them in like green, more minor fencing. The goal is to be able to prevent the escape, because these animals are very strong and very intelligent. And I argue they're much more intelligent than cats and dogs, almost like parrots. Very smart and problem-solving skills, and they're social, and they actually will work together to escape. I've documented it in 5 different breeding facilities. They will cooperate to get out, okay? Tegus, people don't understand tegus till they own them, which is another reason to work with the animal while it's alive, not just trap and kill, trap and kill. There's so much more we learn when understanding this beautiful species, because they are wonderful. So we basically remove them. They come out in these traps. They're usually dumped into a bag. And then what Fish and Wildlife has done is it's like a center punch, what you use to like break. It's similar to what you'd use to break glass on a car that's underwater, like a fireman would use. And so it's used to instantly kill them through the top of the head. Other places have done freezing. Freezing in reptiles doesn't work as well as removing the head, which is why we advocate against rattlesnake roundups for those reasons. It leaves them alive for hours, because cold-blooded, they don't succumb to low oxygen as quickly. So, but the idea is that's the veterinary-approved quick, even though it sounds horrific, it does affect my ability to count scales and do studies on them when I'm working with their necropsies on their faces. Thank you.
Good afternoon. My name's Dave Kraus. I'm here actually learning. My background is in software architecture, aviation, computers, technology. It's not conservation or biology. And I've been sitting upstairs writing notes about everything you guys have said, and it really got me thinking about ways that we can work together. I stand before you as the National Industry Member for Aviation Safety. We are a collaboration facility with the National Institute of Standards and Technology, and I'm a representative of the UAS Center at SBD, which is the San Bernardino International Airport right around the corner. Today we're going to talk about drones and the purpose of the UAS Center and what we do. It's all about economic development, revitalizing the area, stimulating the region, helping each other so that we can collectively grow. Rising tides raise all ships, and that's what we do. In the Santa Ana River watershed, you guys got a lot of issues. Invasive species is something that is fascinating to me. Up until recently, the kangaroo rat was something that I just heard a lot about, and I saw a lot of pictures about it today. Never heard anything about the sucker fish. That was interesting. So I'm learning a lot. And what I already know from some of the work that I'm doing out on the East Coast is how climate change is impacting the environment, and how it is really causing an issue